Chocolate. We love the flavor, but do you know about this treat's dark side? Chocolate is derived from cacao beans grown in tropical climates around the equator. Annual global chocolate production is projected at 7.5 million tons in 2024. As a result of high demand in corporate farming, chocolate production has led to as much as 70% of Cote d'Ivoire's deforestation as rainforests are replaced with monoculture cacao farms, crippling biodiversity. Climate change has led to drastic fluctuations in temperature and precipitation, killing cocoa trees and leading to smaller harvests every year. Chocolate production has also contributed to climate change. The average 40-gram chocolate bar is associated with 5 kilograms of carbon dioxide emissions. And at its core, the chocolate industry is built on unethical, unsustainable practices. From the cocoa farmers earning less than $1 an hour to the 1.6 million enslaved children working on West African cocoa plantations under brutal conditions. While some chocolate brands try to be eco-friendly and ethical, their efforts aren't addressing the bigger sustainability and human rights violations. Most companies prioritize costs, exacerbating the issues. We are the East Coast Bio Crew, a high school team based at a community biology lab, Bugs, in the center of Baltimore. Our project, Lactococcus, aims to revolutionize chocolate production by using Lactococcus bacteria to synthesize 4 key flavor compounds: HDMF, theobromine, dimethylpyrazine, and phenylethylamine. This method is clean, efficient, and ethical, providing a sustainable alternative to conventional chocolate making. Our goal is to produce a chocolate flavor compound that can be sold to large companies, allowing us to collaborate with the existing industry. By providing an alternative to traditional chocolate production, Lactococcus can reduce the industry's environmental footprint, cutting carbon dioxide emissions and preventing deforestation. Additionally, this innovation can create higher-paying, skilled labor jobs, boosting local economies and allowing consumers a choice of chocolate that doesn't involve unethical practices. Ultimately, Lactococcus seems to benefit both the environment and society, contributing to a more sustainable and equitable world. We chose Lactococcus lactis as our host organism because it has a long-standing reputation for being food safe, producing many of the dairy products we enjoy, such as cheese, butter, and buttermilk. We chose our plasmid PNZ8148, which includes a nisin-induced promoter, because nisin is a broad-spectrum bacteriocin produced by L. lactis, used extensively as a food preservative, and thus is also food safe. The combination of PNZ8148 and the use of L. lactis would ensure that our product would pose a lower risk to human consumption. We identified 4 pathways for the production of 4 chocolate compounds: 2-phenylethylamine, theobromine, HDMF, and 3-ethyl-2,5-dimethylpyrazine. This is the signal transduction pathway of nisin. Nisin first binds to NiceK, a membrane-bound receptor protein. NiceK autophosphorylates and transfers a phosphate group to NiceR, which activates the protein. Once activated, NiceR can interact with the pNiceA promoter, activating the expression of the gene. We assembled all of our plasmids using hi-fi assembly. Hi-fi assembly can effectively join together 2 or more DNA fragments using 3 enzymes: 5' exonuclease, a DNA polymerase, and a DNA ligase. We first designed primers specific to each pathway, which would amplify our plasmid with a 5' overhang homologous to the 5' end of the coding region. This 20 base pair overhang is crucial for hi-fi assembly. 5' exonuclease chews back more of the 5' end strand to create a longer overhang so that the 2 fragments anneal together. DNA polymerase fills in the gaps, and DNA ligase seals any nicks and covalently links the DNA fragments together. After the plasmid was constructed via hi-fi assembly, we initially cloned an E. coli and then electroporated the purified plasmid into our final host organism, L. lactis. From here, we plan to induce the nisin promoter with the addition of nisin into the media in order to generate gene expression. We started all our initial transformations in DH5 alpha strains of E. coli. Cloning in DH5 alpha was never consistent in our plasmid. In figure 1, we see visible colonies for each of our genes. However, when performing colony PCR on these colonies, we found that very few contained our successfully assembled plasmid. While 5 out of 7 colonies tested for XMT, MXMT, DXMT plate, and 1 out of 7 colonies tested for TDH-KBL plate were positive, all the other genes were negative. We eventually found out that the reason for the low plasmid stability over generations was due to our selection of a REC-A negative strain of E. coli, such as DH5 alpha. The REC-A gene aids in the recombination of homologous DNA, but is engineered to be dysfunctional in DH5 alpha cells in order to prevent unwanted recombination of the plasmid. However, our plasmid requires the function of the REC-A gene in E. coli for proper amplification, and lack of the REC-A protein can make the chloramphenicol resistance gene specifically susceptible to deletion. So we speculated that we would find a higher success rate in a REC-A positive E. coli strain, such as MC1061. We found much more successful cloning in MC1061 compared to DH5 alpha. Following a 3-day incubation at 30 degrees Celsius, MC1061 E. coli colonies with XMT, MXMT, DXMT, TDHKBL, and FBA-TPI grew. When performing colony PCR on these colonies, 15 out of 15 of all tested colonies of FBA-TPI showed successful insertion, 7 out of 7 of all tested colonies of TDHKBL showed successful insertion, and 2 out of 7 of all tested colonies of XMT, MXMT, DXMT showed successful insertion. The 5 negative bands were likely due to gel loading error. In all transformation attempts in MC1061 and DH5 alpha, no colony PCR attempts on SIPDC, PTPDC, or PGPDC showed successful amplifications. These being the only constructs that code for phenylalanine decarboxylase, the enzyme which produces phenylethylamine. We speculate that due to lack of full nisin promoter repression in E. coli, toxic levels of phenylethylamine or deprivation of phenylalanine could arise in E. coli, killing the cell. However, we still had issues with cloning inside of MC1061. 2 transformation attempts in MC1061 E. coli with assembled plasmids extracted from positive MC1061 colonies yielded no colony growth. After discussing cloning difficulties with the company which provided our team with our plasmid, we were informed of an accidental mutation in the Rep-C region of the plasmid. We hypothesized that this mutation may be the cause of our amplification difficulties and expect more success with the fixed plasmid. When we purified the plasmid from our E. coli liquid cultures, we then electroporated that DNA into Lactococcus. Unfortunately, we were unable to produce colonies. However, with the Rep-C mutation fixed, we will find a greater amplification efficiency for Lactococcus as well. The type of measurement that we've highlighted is analysis of the physical properties of our produced substances to confirm their identity. In particular, we want to emphasize techniques that can be used to analyze scent-producing compounds. Most commonly used is gas chromatography and mass spectrometry, or GCMS technology. We constructed a clear explanation of GCMS technology that explains how GCMS works, how to find a GCMS facility, and how to use it, prepare samples for it, and how to analyze the raw data generated. However, many iGEM teams and community labs such as our own do not have access to GCMS or even any product identification tools at all. We therefore also developed a method for quantifying smells using purchased chemicals. This was important to us because it allowed us to characterize the properties of our target chemicals, providing standards for each chemical we want to produce, and helping us to find the right balance between the different chemicals.
Using our smell test protocol, we characterize the smells of the 4 chemicals by asking each participant to rate the intensity, from a scale of 1 to 5, of specific smells for each chemical. All the chemical concentrations were based on the naturally occurring concentrations found in chocolate. In characterizing the scents of each of the chemicals, we find that HDMF had a stronger average intensity than all the other chemicals, with an average intensity of 2.43 compared to around 1.6 for all the other chemicals. So, reducing the concentration of HDMF in further experiments will likely be necessary to ensure the right balance of chemicals needed to resemble the chocolate smell. This experiment was also essential to establish standards of odor for each chemical. These standards allow us to create controlled experiments in the future when testing chemical ratios and assessing the purity of our biosynthetically produced chemicals.
Through human practices, we gain valuable insights into how our project could impact the wider world by interviewing industry professionals and surveying the public. We met an FDA food scientist to explain that our product, classified as a precision fermentation, would require FDA approval for market entry. Dr. Mark Giltonen, a Pennsylvania State University botanist, suggested that we consider multiple approaches towards the effect of our project on cocoa farmers, making a concerted effort to consider the positive and negative consequences. We also spoke with Slave-Free Chocolate to better understand the complicated topic of child slavery in the context of cacao farming. Their advice to focus on education surrounding the issue pushed us to reach a broader audience with our outreach campaign of seminars and hands-on workshops. Symbio for All Africa told the progress of synthetic biology education in Africa and sought advice on the risks and benefits of implementing synthetically produced chocolate. Irvin So, a scientist at McCormick, gave insightful advice on scaling up the production of synthetic chocolate and ensuring its integration into the flavoring industry. As a way to engage with the public, we conducted two surveys, one with biased wording critical of current cocoa production practices. Statistical analysis, like chi-squared, help us determine the effect that biased wording had on whether people said they would buy synthetic chocolate, and identified ethicality as a concern. We ran 3 tests on variables such as whether ethicality was selected and whether people were more likely to buy synthetic chocolate. We also discovered that most people were unaware of chocolate production issues, indicating a need for more education. Additionally, respondents did not favor ideas like scent or dog-friendly chocolate, so we chose not to pursue those. We then held 3 focus groups with 4 to 5 participants each to explore survey responses further. We focused on why ethicality was or wasn't a concern and what ideal chocolate production looked like to them. We also gauged community members' comfort with synthetic products, factors that might attract or repel them from synthetic chocolate, and their views on government regulation. 2 key themes emerged. First, participants were generally open to synthetic biology or genetically modified products if they provided benefits, especially in addressing labor and climate change issues in the cocoa industry. Second, the biased survey wording did indeed influence whether people selected ethicality as a concern, showing how wording can impact responses. Our education subteam hosted workshops and speaker sessions for children and adults in the Baltimore area to educate them about different aspects of our project, including cocoa harvesting and chocolate production, impacts of climate change on food systems, sustainable food production, and genetic engineering techniques. For our kids' classes, we planned fun and interactive presentations and activities to both engage and educate them about these topics. These included a spherification activity to learn about food science, a chocolate tempering technique to learn about chocolate's properties and the production process, a bacterial transformation lab to teach kids about synthetic biology techniques, and activities related to sustainable food systems. We also had speaker sessions for our team members and adults to learn about these topics, including Dr. Giltonen from Penn State University, who presented about cocoa genomics and supporting the future of chocolate and cacao farming, Vivian Su from Johns Hopkins University, who presented about alternative proteins and sustainable food production, and the Reed Center, who presented about sustainable farming. In total, we were able to reach around 100 parents, children, and community members through these public sessions and workshops in order to provide greater understanding of our project and its support in multiple different areas. In addition, we used Instagram to share small facts about chocolate. We called these bite-sized bits of information chocolate chips, and they were among some of our most viewed posts. Our project focuses on targeting large companies that buy in bulk. Our Cacao Alternative Report outlines guidelines for farmers that will help produce less labor-intensive crops. This shift can reduce the reliance on child labor. Our vision for this project begins with safety, with our FDA guidebook planning our journey to FDA approval. This guidebook will also pave the way for other community leaders aiming to launch projects of their own to U.S. markets. Next, we'll work with stakeholders to distribute the technology ethically, whether by founding a business, partnering with organizations like Symbio for All Africa, or selling it to companies. We are also aligning our efforts with several key Sustainable Development Goals, or SDGs. We tackle SDG 9 using a holistic approach by providing the chocolate production industry with sustainable alternatives, as detailed in our Cacao Alternative Report, or CAR, and by supporting innovative food science projects in the U.S. via our FDA guidebook. We're focused on large companies that control nearly 60% of the global market, ensuring that traffic shifts towards sustainable options. Thanks to our CAR, we've also set up a safety net for farmers who might be impacted by this transition. For SDG 12, we emphasize a commitment to responsible scientific production, both through technological support and educational outreach in the developing countries our project will impact. Working and discussing with organizations like Symbio for All and Slave Free Chocolate, we promote sustainable synthetic biology solutions that reduce environmental harm and foster economic growth. Additionally, we worked with experts from McCormick to learn about scaling up synthetic chocolate production. For SDG 15, our efforts to replace traditional cacao farming with less resource-intensive synthetic alternatives will reduce deforestation, supporting the preservation of land ecosystems and biodiversity. We focused on educational outreach, providing seminars with experts such as Dr. Mark Giltonen and classes on synthetic biology.